Okay.
Hello. Oh. Good afternoon, or whatever time it is where you're watching, and welcome to the third plenary session of this H3 Africa Consortium meeting. Without more ado, I'd like to introduce the first speaker, who's Benedict Payton, and he's going to be talking to us about the Human Pangenome Reference Centre. So over to you, Benedict.
Project of the National Human Genome Research Institute of the NIH. So, as many of you will know, roughly 20 years ago, we released as a community the first human reference genome in a series of papers in 2000. So there was the public effort, as many of you will know, and there was also a commercial effort to try to sequence human genome. And in the end, actually, both efforts learned from one another, and the reference that we have used for the last 20 years is really a result of this initial foundational work. And in fact, fast forward to February 2021, and there was recently, a paper there was an issue of Nature celebrating that 20-year landmark achievement and all the things that have come from it. But the human reference genome actually is a single haploid representation of a human genome. And as such, it does not adequately represent all the genetic diversity that is present in the human population. If you think about what we use the reference genome for in genetics, it's pretty startling, right? We use it as a basis for viewing genes, for viewing promoters, for thinking about the way in which our genome is organized. It is a coordinate system for everything that we do in genomics today. And increasingly, it's used in a clinic as a reference structure for the purposes of assays and so forth. So when you go and get a genetic test, that reference genome is implicitly used, and I'll get a little bit to that towards the end. But it is actually in the way it was constructed, it's actually a chimera of several different individual samples, although mostly one sample, 70% one sample. And because it is really just one haploid representation of one human genome, it cannot be a representation for all of humanity. So, fast forward to 2020, and the Human Pangenome Project was kicked off, which is essentially an effort to try to build a better representation, a better public representation of the diversity of the human population, and to make that, in the same spirit of the initial Human Genome Project, to make all of that data free and publicly available to everybody to use as a basis for future genomics projects. And this project has the aim of sequencing at very, very high quality, so at reference level, at least 350 diverse human genomes to build a comprehensive map of human genome variation. And in the process, to actually create a whole bunch of new standards that are necessary to reason about variation at the scale of hundreds of individual haplotypes and how they interact structurally. So what does it look like? Well, here's the sort of cartoon version of this that I'm going to come back to multiple times during this talk. So we imagine taking our diverse set of samples contributed by individuals from around the world, and we sequence each one of those samples, and therefore each sample gets represented as a pair of haploid genome assemblies, right? One representing the paternal assembly. And here in this picture, I'm representing them as kind of colored rectangles sort of streaking along from left to right. And we can imagine that wherever those sequences are the same, and of course, because any two human genomes are startlingly similar, whenever two genomes are the same, we're going to merge them together. And then when they're different, we're going to allow them to diverge. And in so doing, we no longer have, a set of linear strings, as we've been used to dealing with the human reference genome. We actually have a mathematical graph where divergences create bifurcations that define a structure that sort of weaves variation implicitly within it, and that's what's shown here. Again, I'll come back to this a little bit more towards the end of this talk, but you can see that we have a mathematical graph structure in which we have nodes that represent individual snippets, individual sequences of DNA, literally strings of A, T, C, G, and then we have connections that indicate the way in which those snippets of DNA are actually organized in an individual haploid genome. And those connections actually represent individual haplotypes as they flow through the graph. So you can see, for example, here that we take this red genome here, we can actually trace the fact that it initially comes through this GAT sequence and then goes through this GCTC sequence and so forth, right? And you can see that the haplotypes diverge here and so on. So that is the overall, very, very high level vision of the project. But there are a number of technical challenges to get from where we are today to that vision of having a comprehensive, public reference structure that we can use as a basis for genomics. And one of the big technical challenges that we faced over the last 2 years or so is this production of complete human reference genomes at a quality that is equivalent or better, actually, than the current reference genome. So one thing to mention about the human reference genome is that it never was complete. So GRCH38, which is the current human genome assembly that most groups use, or flavors thereof, is actually only about 92% complete. There's about 8% of the sequence that is not actually haplotype resolved, right? And that is because, when the Human Genome Project was finishing off in, 2003, the aim was actually just to try and finish all of the euchromatic regions of the genome. So sequences that were centromeric, the acrocentric arms, the telomeres and subtelomeres, and within some of the more duplicated satellite arrays and some of the segmental duplications, those were never intended, or at least it wasn't envisioned that they could be finished in the initial human genome assembly. But of course, 8% is a significant amount of our genome, right? So what's going on in those regions? So the Human Reference Genome Project, Human Pangenome Project, is not just about trying to create more genomes, it's actually trying to create more complete genomes, so-called telomere to telomere genomes. So, okay, so how are we organized as a project? Well, actually, to try and do this and bring all of these aspects together, we've kind of leveraged multiple different groups and multiple different sets of expertise. So we have a group that is focused on population sampling and representation. So how do we select samples from around the world? Given that we have a limited budget, we have a limited number of samples that we can do, how can we maximize the amount of diversity that we capture so that most people are as represented as best we can at this stage of the project? So that there's a lot of population genetics representation in that group. And then there is a sequencing technology group that is really focused on then translating those samples into digital representations of DNA. And for that, we have very able groups that are using a variety of different sequencing technologies that I'll get to in a second, to allow us to build these genome assemblies. And then the groups that I'm really mostly involved in are the groups for actually constructing the genome assemblies from that sequencing data, and then having done that, building the pangenome itself and all of the underlying data management that is necessary to make this quite large bolus of data available to everybody in a free and fair way. And underlying all of this, I should emphasize, is an embedded ethics and policy working group that is trying to provide oversight so that what we do as a group does not repeat the mistakes of the past, and does so, and uses samples from individuals in a responsible and ethical manner. So, okay, so what have we got? How is the project going so far? So, in our what we called our first year freeze, we focused on just over 30 genomes, and this is and we selected those 30 genomes from a pool of actually samples that come from the 1,000 Genomes Project. So we chose the 1,000 Genomes Project initially just because those samples are available to us. They have been banked at Coriell, and we could get high quality DNA. And so we took the 1,000 Genomes Project, and then essentially we looked for lines that were part of trios, so that we had, mother, father, and offspring. And that's really important because having trio information at this stage allows us to create better genome assemblies, because it allows us to pull apart the maternal and paternal contributions to the child's genome. We also looked for lines that are low passage, so to minimize the number of acquired mutations that are derived during the creation of the cell lines and subsequent generations. And of course, we wanted to maximize genetic and geographic diversity, and that goes back to that sampling group. So we have, this is basically the set of data that we had, the set of available lines that we had to choose from for our initial first and second year sequencing at this point. And you can see that it represents, a reasonable number of different subpopulations from around the world. But we know that we need to go, if we want to reach our targets of 350 plus reference genomes, we know we need to go beyond 1,000 genomes, which, whilst diverse, was not actually, there are still plenty of underrepresented groups within that were not sampled by 1,000 genomes. And so we are really trying to reach out and create partnerships to create additional samples and additional genome assemblies. And as such, we've kicked off an alliance, overusing the word alliance, a partnership, I should say, with the Global Alliance for Genomics and Health to try to bring other groups that might be interested in constructing these really, really high quality reference genome assemblies, so that we might, define common standards for genome assemblies, but also for data sharing protocols, and for consent protocols, and the ethical considerations and so forth, and then ultimately to integrate that data into this reference pangenome structure that we're building. And if people are interested in that discussion, I will attempt to represent the group. That's actually not my group, but I will certainly be able to funnel people towards the right people to talk to try to get those partnerships going. How am I doing for time? Okay. So in order to create the sequencing data for all of this, we've actually created a kind of virtual sequencing center across a number of centers of expertise within the United States, including my own institute, UC Santa Cruz, but also University of Washington. There is also efforts, not sequencing efforts, going on at the European Molecular Biology Lab, and then at Rockefeller and at WashU. We have sequencing going on. So essentially, this is the distribution that is creating. So all the nanopore sequencing is being done at UC Santa Cruz, and then most of the PacBio sequencing is happening actually at University of Washington and WashU and Rockefeller. So what are we doing in terms of sequencing? Well, we are producing the workhorse of the project right now. Is this PacBio high fidelity, so hi-fi data, which is this new technology, relatively new technology from PacBio that uses this circular consensus technique. So what that essentially means is that they create a circularized molecule, and then they run it through the PacBio instrument multiple times, essentially iterating around the circle multiple times, and then they basically align all the copies together and derive a single read that has extremely high consensus accuracy. So typically kind of Q30-ish, so one error every 1,000 or so bases. And for this project, we're generating, in general, at least 35x coverage of these reads. And they typically have a read length of about 18 to 20 kilobases. And then to sort of complement that, because 18 to 20 kilobases is certainly long compared to an Illumina read, we also are using ONT, so Oxford Nanopore Technologies, ultra-long sequencing. So this is read molecules that have a lower accuracy. They're perhaps 95 to 97% identity. So, you know, one error every 20 or so bases, so a lot less accurate, but significantly longer. So here's the sort of a stacked bar plot showing you the distribution of reads less than 100 kilobases, and then 100 kilobases to 200 kilobases, and then 200 kilobases plus. Here's a sort of throughput distribution. And so typically, this allows us to get for each sample a healthy dose of reads that are 100 kilobases and 200 kilobases long. And that essentially can be used to scaffold together and finish off the gaps that the shorter PacBio Hi-Fi reads can't walk through. So these two technologies are extremely complementary for the purposes of really high quality genome assembly. Yeah, so we've actually made a year one sequencing release for our first 30 samples picked from those 1,000 genome set. And we have for pretty much, not quite, but pretty much PacBio, certainly PacBio Hi-Fi and Hi-C, which is this proximity ligation technology from Dovetail, as well as BioNano and Illumina for all the samples, and then StrandSeq, which is this really cool technology that allows us to distinguish which strand the reads came from for 10 of the technologies, and it's particularly good for studying inversions. And then we have an additional 12 samples that we're also including that include a subset of these technologies. And we've made all of this data available for public download for free from this Amazon Public Datasets project, so this S3 URL, Human Pangenomics, and also through the NHGRI Anvil project. We have a workspace, which again is publicly available. You can go in there and you can actually play with the sequencing data. You can download samples, or you can literally on the Anvil analyze those samples and do analysis. And we've made all the workflows that we've built for actually creating the assemblies and doing the QC available through the DocStore project. Okay, so very briefly, because I know I don't have too much time, in terms of how we've built the assemblies, I won't take you through this whole pipeline, but the workhorse for this year one has been the Hi-Fiism assembler. So after doing some initial cleaning of the reads to remove adapters, we run this trio Hi-Fiism, which takes in the Hi-Fi data plus the Illumina data, the Illumina trio data that allows us to pull apart the maternal and paternal chromosomes. And then post-assembly, we do a number of cleanups to filter any decontamination contigs, to remove any dimers, and then there's a few things to pull out the mitochondria and so forth and so forth. Anyway, all the assemblies now have been actually uploaded and accessioned through GenBank, and this is an old slide, but this is all now available. And I'll show you the stats on this in a second. So yeah, so the Hi-Fiism program was selected after we held a benchmarking competition. We have a paper coming out that explains this benchmarking competition, but basically we asked a whole bunch of the sort of best assembly groups in the world to build the best quality assembly for the HG002 benchmark sample, which is this Ashkenazi Sun sample. And we got, for using Hi-Fiism, we got an assembly at the time with an NG50 of about 43 megabases with phase blocks sizes of about 18 megabyte bases, the N50, and a Q50, so that's one error every 100,000, a little bit less than one error every 100,000, and a very, very high accuracy in terms of just the overall retrieval of het SNPs. So yeah, I won't take you through, but this is now published, and it's a really, really nice method that has become sort of our workhorse within the project. And then post-assembly, we have workflows to basically run QC on all the samples to check their quality. So we're looking at the quality of the variants that they produce, the quality according to k-mer analyses, quality of read-based alignments and so forth. and we make all of that. We make all of that available as part of our release with each and every sample. So you can actually go in and look at the assemblies, and then you can look at the QC statistics for each assembly, and you can pick the samples that you like the best. So for example, just to show you this, in each one of these, this bar plot just shows you the length of the assembled sequences, so concatenating the assemblies together for each sample. And in orange, I think you've got, I think it's the maternal assembly, and in blue you've got the paternal assembly for each sample. And you can see that basically each assembly is roughly 3 gigabases in length, so which is pretty close to we think most genomes are roughly 3 and change gigabases in length. So the vast majority of sequence is apparently assembled. And then the number of contigs, it varies a little bit. But typically each assembly is, these are contig level assemblies, they're not scaffolded. Each assembly is in roughly 400 pieces, which is pretty good. Yeah, in terms of the quality, all the assemblies are around Q50-ish, so as I say, about 1 error every 100,000 bases. And the N50s are on average about 40 megabases. So your average contig length, 50% of your, I should say, 50% of your assembly is in contigs that are 40 megabases or longer, which is pretty amazing. Yeah, very low, I won't drag you through this, but very low levels of misassembly in terms of apparent switch errors between maternal and paternal, and then this is another measure of errors in terms of phasing. One of the things that we've done to measure the accuracy of the assemblies is to realign all the reads back to the assemblies, and then use a sort of mixture model deconvolution to try to find regions where we see more reads aligning to a region than we would expect. And those represent regions of the assembly that are potentially collapsed, where the assembler has not actually created sufficient copies of that region. And so it's sort of made a sort of collapsed representation. And then, of course, regions where reads do not align reliably, which are potentially regions that have been noisily assembled. And we actually find, doing this analysis, that more than 99% of every assembly is well supported by either the hi-fi or ONT reads. So this is percentage, this is 0.3%. So you can see there's a very, very low rates of unreliable blocks in our assemblies. And when we look at where the unreliable regions are, they're almost all in either seg dupes, centromeres, or a combination of both centromeres and segmental dupes. So the errors that we're making are in the most difficult regions, the satellite arrays, essentially, of the genome. So pretty much all the genes and so forth are essentially perfectly assembled. Okay, again, I want to not finish on time. I want to finish on time. So where are we going now? So having built this initial set of assemblies, which are now publicly released, we are now in year 2 of our assembly process. We're adding an additional 50 assemblies quite soon. That will be another 100 haploid assemblies to the mix, and so forth. That will continue to grow over the years. But what we now have to do essentially is take those assemblies and integrate them into a pangenome. So what is a pangenome? You can think of a pangenome as that set of genome assemblies plus the alignments that relate them, okay? So it's assemblies plus alignments, and it allows us to then view all the variation that is common between the samples. And to do that, we use these things that I introduced at the beginning, these genome graphs. And the reason we use genome graphs rather than traditional multiple sequence alignments that you might view in a matrix is because matrices can't represent the kind of complex rearrangements that take place between genomes, like inversions and duplications and so forth. So genome graphs are essentially just a necessary structure for representing the alignments between assemblies, nothing more. And we have collectively built now and are testing 3 different pipelines for pangenome construction, because this is definitely an area of active research. And we have a pipeline from Heng Li's group, the minigraph pipeline, which is actually published. And then we've developed the UCSC group, so my group with Heng has developed a hybrid pipeline, this minigraph cactus pipeline, that essentially builds on Minigraph to add in a lot more detail. And then from Eric Garrison's team, there's this pangenome graph builder. And all of these toolkits are to some extent available. And we're in the process of creating a marker paper that will publish the year 1 assemblies and these initial pangenomes and demonstrate what we can do with them. Just to sort of show you what that looks like, within the UCSC browser, we're creating visualizations of the pangenome. Here, for example, is a UCSC browser screenshot showing you some genes. This is this TRIM for 49T1 gene, and you can see here that most of the sample, or some of the samples, agree with. Here we're sort of projecting the pangenome back onto GRCH38. So the coordinate system here is the reference, current reference human genome, and you can see that many samples agree with GRCH38, but a significant number of samples actually invert this last exon of TRIM49D1. Right? So it's this kind of information that the pangenome gives us, this kind of resolution, as well as giving us indications of SNPs and so forth, and the haplotypic variation through that region. And what's cool about these browser visualizations is actually that you can surf to any one of those genome assemblies and view all the annotations and so forth from the perspective of that given reference genome, which I think is the future. I think being able to view human genetics from your reference is a really exciting possibility. So there are lots and lots of technical applications of the pangenome that we are in the process of demonstrating through a whole series of publications. One of the key things is just improving genotyping. So how do we infer the next genome given our knowledge of an initial pangenome? And we have papers coming out showing the genotyping of with both short and long reads, and the genotyping of structural variations with both short and long reads. We're also looking at phasing and imputation, and a whole bunch of other things to do with transcriptomics, ancestry inference, visualization, which is a key one, and things that you can do with k-mers and k-mer-based approximation methods. So lots and lots of things to come on the sort of technical side of applications with the pangenome. And then just finally, as I said at the beginning, this isn't just about creating more reference genomes, it's about creating more complete reference genomes. And sort of in partnership, the Human Pangenome Project is working with Adam Phillippe's group, NHGRI, and Karen Meager, who's my colleague at UCSC, to create these telomere to telomere genomes. And so the aim is for at least a subset, ideally all, of these 350 genomes will ultimately be completed to telomere quality. Of course, the issue with that is that we don't yet know how to do that at scale. But we have released, this is again all Adam and Karen, but the Telomere to Telomere Consortium has released the first complete telomere to telomere genome assembly, where literally the assembly is just 23 chromosomes. There's no Y yet. There will be very shortly. And there are 0 unlocalized, 0 unplaced contigs. The assembly is, as near as damn it, complete. And there are only 11.5 megabases of ends, which represent the rDNA locus, which is still unresolved, but in the process of being fixed. And one error every roughly 10 million bases. That's an extremely low error rate. So yeah, this is now all available. There's a whole series of papers that have been submitted now and are available in preprint that describe just the incredible biology that's being discovered through this. And so this is very much a partnership. And where we want to go with the Human Pangenome Project is ultimately to achieve this level of assembly, but for our 350 diploid genomes. Okay, so just to wrap up, we're not there yet. We have this initial haploid telomere to telomere genome. The next step will be, creating a diploid telomere to telomere genome, and that's really the focus of the group now, the assembly production group. And then finally, how we get to hundreds of these things and integrate them into that pangenome. So, yeah, I'm out of time, but when we're done, we hope that what we've created will have implications for healthcare. It will open up more of the genome and more of the genetic diversity that exists in our population for biomedical applications. We hope it will help us discover new things about genomics, and we hope it will help us understand our collective diversity better. And there's a huge number of people to thank here. I want to especially thank Karen Meager, who helped produce a lot of these different slides, and the whole of the Telomere to Telomere Consortium as well, which is this sort of parallel group that's working with us. And I will stop.
Thank you, Benedict. That was a fantastically informative introduction to this session, and thank you also for being conscious of the time. We've already got questions coming in. I'd like to remind the audience that they can post questions in the Q&A box, but we're going to hold those for a moment, and we're going to have the second talk because it's on a related topic. So we're going on to Sean Aaron's talk on the H3A Bionet African Pangenome and Reference Graph Project. And then after that, we'll have a joint Q&A on the first two talks. So over to you, Sean.
Okay, great. Thanks, Ruth, and hello, everyone. So as mentioned, my name is Sean Aaron, and I'm a member of HA Bionet. And firstly, I want to thank Dr. Patton for his great talk and providing an insightful overview of the HPRC, as well as setting a lot of the foundational knowledge about genome graphs and pangenomes. So I'm going to be continuing with this topic, and in this talk, I'm going to provide an overview of the HA Bionet's African Pangenome and Reference Graph Project. So just to recap, reference genomes, as we know, are an integral component of genetic studies, and they form the basis for identifying genetic variation in newly sequenced genomes. And as mentioned, the most commonly currently used reference genome is GRCH38. And as was mentioned in the previous talk, while it was considered complete, it still has several unresolved and complex regions that are yet to be accurately sequenced. As mentioned as well, one of the great initiatives is the Telomere Consortium, was managed to create or produce the first complete telomere-to-telomere high-quality assembly of all human chromosomes. But while this has been a great achievement, there are still some limitations in terms of that assembly, in the fact that it is still haploid, and it still does lack diversity. And as a result, it may not be, extremely accurate at representing population-specific variation. And as we know, a lot of this population-specific variation that we observe in the genome does occur in some of these more complex regions where there is a lot of structural change or structural variation. So I'm sure everyone in this audience will appreciate and be aware that African populations harbor the greatest genetic diversity, and unfortunately, it hasn't been fully explored and identified, and is not well represented in a lot of public genomic resources. And this is particularly true for the current linear reference genome, which fails to represent, a lot of genetic variation, which occurs in a variety of minority populations. And so I'm showing you recent results from one of the HA Africa papers, where we looked at whole genome sequencing in just over 400 African individuals. And what we were able to see here in terms of diversity, that while there seems to be a continuum of diversity across the continent, we do see sort of this distinct geographical variation across the continent. And you can see there PC1 in this principal component sort of separating our populations from west to east, and PC2 separating our populations from west to south. And so much of this variation we know exists because of the complex history and differential admixture that occurs in Africa, and admixture with local groups, as well as complex migration and demographic events. And so the extent of genetic diversity is great. And I think, and that's what makes it quite challenging to be able to identify a lot of this diversity when comparing whole genome sequencing to a reference genome that is not as diverse and representing the diversity that does exist in these populations. So just in addition, in that study, we were able to also identify, that there is this great diversity in Africa, and there's a number of novel variants in African populations, and that number has not plateaued yet. And this is just showing in the graph the novel variants that are common with a minor allele frequency of greater than 5%. And you can see that even in these last two populations, we were able to identify Just between those two populations, additional 6,000 variants that we looked at that were identified as novel. So this genetic variation that we do see in African populations is large. And it is a challenge to represent not just single nucleotide variants, but structural variants in particular in a linear reference sequence. And there is a need to generate a much more specific African reference resource. So while one of the limitations was or has been a lack of the availability of whole genome sequences, we do have a reasonable amount of sequence data available on the continent now. And this image just shows you a subset of the sequencing data that's available. And in addition, which was clearly showed in the previous talk, we now have an improvement and advancement in technology, sequencing technology, that has allowed us to produce, really high quality, accurate long read sequencing. And coupled to that, we've had the development of these highly efficient algorithms that have allowed us now to produce high quality de novo assembled reference human genomes. And so we're now at stage where population scale building of genomes is a reality. And we can use this to generate much more population or regional specific reference resources. So I just want to give you an overview of the project. So these are just the three sort of main overarching aims of the project. So the first is to use available short-range sequencing data to try and identify African-specific genomic sequences, identify these sequences and characterize them, and then use that to build a linear reference pangenome as it's coming from multiple individual genomes. The second part of the study is to identify or to generate long-read sequence data from diverse African populations and use those long-read sequences to generate an African-specific reference genome graph. And then ultimately to try and evaluate, the accuracy of using these reference resources to call variants as opposed to using the linear reference sequence. So this is just an overview of the pangenome approach that which is part of the study. So we're starting off with a data set of about just over 400 representative African whole genome samples. And these are short-read sequence data sets. And in the table, I'm just showing you the different groups that we have. So again, we've tried to capture that variation in terms of the different regional populations that we have access to. So ideally, we're going to use the whole genome sequencing data sets. We want to identify and extract reads that do not map to the current reference. So current reference that we're using at the moment is GRCH38. We want to extract those reads, assemble those into larger contigs, and then try and look at annotating those reads, those contigs, and where possible, map them back to the reference genome to see where they might map back to. Obviously, an important part of that is trying to characterize, these contigs that we identified as being specific to African populations, and then also to examine the contribution, so the, which regional populations are contributing to these African-specific contigs. And to do that, we're using some established workflows that have been developed. So there's the UPAN pipeline that has been developed to do this type of analysis. And we've also developed an in-house workflow, which is based on the Sherman et al. Paper, which tried to examine African-specific contigs, but in African-American individuals. And we're building these two workflows in order to generate data using both of them and compare what we get from both these workflows. And so we're using a combination of H3 Africa data. So you'll see that there's some Baylor data sets in there. So those that have been generated as part of H3 Africa. And then we also are using some data sets that have been generated from public resources. So in terms of the pangenome progress, we've set up some workflows and tested it with 1,000 genomes data. We've run into some issues that we managed to troubleshoot. We've developed some annotation approaches for downstream analysis. And we also are now investigating how to incorporate the T2T CHM13 reference genome into the analysis. So we've managed to de novo assemble our genomes. We've run some QC and we've removed some samples removed due to contamination. That's a lot of the public data that we're using. And we're currently running our samples through a UPAN workflow. And again, we're doing this analysis per ancestral group. Again, we want to identify the specific contributions to these African-specific contigs, and also evaluate the effects of read coverage, because we're using both high coverage and medium coverage data. In terms of the reference graph approach, again, here we wanted to select samples in order to represent these different ancestral components that we've identified based on the short read data. So what we've done is we've managed to try and source samples to represent these different ancestral components, which are represented in the different colors, which correspond to the admixture analysis that we generated in that study. And in terms of sample selection, we've gone back to a lot of established HA Africa projects, and we were very excited that that PIs within those projects were willing to get involved and contribute samples to the study. So we've managed to get representation of the ancestral populations that we wanted to include. And then just to mention that we also entered the INCABA competition, and we won 3 additional long-read sequencing data sets. And then in addition, we're using some 10x genomics assemblies, particularly for rainforest hunter-gatherer samples and sand samples. And we're hoping to get some additional khoisan samples as well to sequence. So in terms of the approach, so consent has been a big issue with us for this project, because while a lot of the HA Africa samples have been generated for broad consent, we wanted to make sure that we get consent to generate a public resource, because we will be wanting to generate a public resource from this particular study. Our data set, we're using between 25 and 30 samples. We're going to generate some high quality long read data sets using particularly generating PacBio hi-fi reads. We're going to use those to produce high quality pseudo-phase de novo assemblies, because we're not using trios. And we've been looking at hi-fi ASM and high-K-NIT to do that. And ultimately, we want to use the de novo assembled genomes to build an African-centric genome graph. Again, Benedict mentioned some of these tools that we'll be using, and we've explored already setting up these tools and running them on some test data. And ultimately, we want to use the African genome graph to call variants and short read data, again, short SNVs as well as structural variants, and compare some of the results that we get based on the linear reference sequence, so compared to the linear reference sequence. So in terms of progress, we've sourced the samples for sequencing. Ethics approval has been a bit of a challenge, and we've managed to get over that, and because we wanted to be clear about what we're going to be using the resources for. We've managed to get over a lot of the hurdles there. There's 1 or 2 that we need to get through. We've investigated a lot of the tools, and we've developed some workflows for de novo assembly and graph building. And then I think this was also mentioned in Benedict's talk, but this is a rapidly evolving field, and the tools are being developed, as we're going along. And so we've actually, we're in the process of, forming this collaboration with HPRC, where we have been attending some of the working group meetings. And the aim there is to make sure that we align ourselves with sort of the processes and tools and workflows that they're developing for their analysis, to make sure that we are sort of aligned with what's been done in that group, and with the idea of also being able to share some of our data at some point, if necessary, with the HPRC. So ultimately, for the project, we want to generate these 2 resources and be able to recall some short read data, both single nucleotide variants and structural variants, and compare that with what we've got using the linear reference genome, and hopefully we can improve on that, in particular looking at structural variation. Then we also are interested in annotating and characterizing some of the novel underlying contigs that we get out, particularly from the different African regions, and novel contigs that are novel to African populations. And then ultimately, we want to create this public African-centric genome reference graph that will hopefully be a resource that we will be able to use by others in order to more accurately call variation in African data sets. So with that, I'd like to complete by acknowledging that this is a project that's ongoing within HCA Bionet, and it's a number of people involved in the RefGraph work package, and then to acknowledge the funding for HCA Bionet. Thank you.
Thank you very much. We've now got time for a short Q&A session. Could I just remind everyone, by the way, I mean, most people have done this, that when you post a question, it's a good idea. In fact, we're asking you to identify the person to whom it's directed. Now, I'm going to start out with a question for Benedict, and then have one for Sean, and then go on to others. We probably won't be able to get through all the questions that have been posted, but they can still be answered after the session, or even during the session in the live Q&A box. So let's start with the first one. And Jennifer Troyer has a question for Benedict. How much does a high-quality telomere-to-telomere genome cost now?
That's a good question. If I'm not sure what the total cost for the CHM13 assembly was, but it would be in the tens of thousands. But if I were doing it again today, and knowing now what we know now, we're talking about of the order of 10,000-ish in terms of sequencing cost to generate the ONT and HiFi data sets. And then a rather larger amount of money in terms of person time to actually get the assembly done. And the reason that I say that it's large at the moment is that there is a manual step to generate that telomere to telomere assembly. So having initially constructed an assembly graph, which essentially represents the sequences, and then where you're not sure, kind of like, things merge together, sort of tangle together. To resolve those tangles has been a manual process to date. That's how CHM13 was done. But the work now is focused on automated tooling that can pull apart those tangles, automatically. And when we get there, then we will be able to, and I think we'll be at the sort of 10,000-ish cost to get that deployed telomere to telomere. But that is not reality today. I just want to emphasize that no one has yet constructed an automated telomere to telomere assembly, let alone a deployed one. But it will happen over the next year or two.
Thank you very much. Right, a question for Sean. How will the African pangenome be shared and used? Will it eventually be integrated with the global pangenome reference?
So thanks for that question. I think that's why we sort of have reached out to HPRC, or they reached out to us, is that, in the long run, to have a well-representative reference graph, we want to have as much variation represented in there as possible. So ideally, while we will run, or we are running this, running and doing this project on our own, the idea is to have it incorporated into the larger project at some point. There are some challenges around that, as in, we're looking at it from a somewhat simplistic or simpler point of view. So the HPRC is generating a lot more data than we are in terms of what's required to create these high quality de novo assemblies. So I mean, there are some complexities around that. But yes, definitely, it's better that it would be incorporated at some point into the larger endeavor. Yeah.
Thank you. Well, the next question, I think you may both have a comment on. It's from Michelle Skelton, and it's: would an ancestral or more contemporary pangenome be more useful for solving human health issues? Benedict, do you want to go first?
So when you so it's a little unclear on the implication. Certainly, as we organize different human genomes together into a pangenome, one way to think about it is that at the root of all of, at the root of the coalescence of all human genomes is this ancestral population that you could construct a reference genome for, and that would represent a sort of ancestral set of contigs that we all descend from. It's not a single genome because there is no single point in time when we all coalesce at the same moment, but it is nevertheless, there is this sort of ancestral chimera that you could put together that would represent the common coalescence of every human being on the planet. And that population existed roughly for most parts of the genome about 1 million years ago, is my understanding. So we certainly are trying to organize our algorithms so that they use evolution and that sort of ancestral history to organize the data and make it parsimoniously organized, which means using evolution in an intelligent way. So yeah, that it is very much on our mind to try to make that ordering. We are explicitly not trying to put together events that took place further back in time than our common ancestor, because we're interested in all of the variation that lives within our population, not that longer range history that goes back, many, many, many millions of years, if that makes any sense.
Absolutely. Sean, did you want to comment on that one?
No, I don't think there's anything much more. I think Ben is pretty much covered. I think it is something that can be, it is something that can be done with the data, and it is something that we should be considerate about.
It's worth saying that many of the African genomes will be interesting in that putting together that ancestral history because of the human population history and the bottleneck and so forth for out of Africa population. So there is, it's vital that we have all the African contribution for reasoning about recent human history, obviously.
Yeah.
Okay, well, we now have a question from Michelle Ramsey for Benedict. And it says, ideally, one would like 2 haploid telomere to telomere genomes for each individual. You have now done that for 30 people with more coming. How do you then have a single human pangenome reference sequence that represents all genetic variation in humans? Will it essentially be an annotated graph genome?
Yeah, so a couple of things. So first off, we have not created 30 telomere to telomere genomes. We've created 30 very high quality genomes, but they are none of them yet telomere to telomere, with the exception of CHM13. CHM13 is the only telomere to telomere human genome assembly yet created. We do have contigs that span through centromeres and telomeres and so forth, but getting them to be validated and finishing them is going to be a pretty big effort. So just to clarify that. In terms of, yes, where we're going is ultimately exactly what the questioner asked, which is to put together these telomere to telomere, phased genomes for each individual. But yes, exactly right. Once you take them and you squash them together where they are the same and allow them to diverge where they are different, then you have a mathematical graph. All that graph really says is it's just a coordinate map that tells you how one genome relates to another genome. When we actually do analysis, we can always, as I tried to show with that UCSC browser screenshot in the talk, you can always project back onto a reference of choice. And I think that's important because in the long run, we can imagine individual reference genomes, individual personalized genomes being used as the basis for display. So we can always take the data, project it through our pangenome, and derive a linear version that represents one genome or one viewpoint of the data.
Great. Well, thank you very much. I think we'll have to wrap this particular part of the session up now, but could I ask Benedict and Sean to have a look at the Q&A box, and you can answer any remaining questions using the answer feature. So thanks a lot. We'll now go on to the next set of presentations, which are all concerned with biorepositories. And the first speaker is Elizabeth Main, who is going to talk about the biorepository at CLS. Do we have Elizabeth ready? Yes.
So good morning to HE Africa. I'm going to be talking a little bit of a different topic this talk. I'm going to be reflecting on some of the lessons we've learned over the pandemic over the last year and a half with respect to biobanking and how that's been impacted by the COVID pandemic. Just a reminder that there are 3 biobanks attached to the HE Africa Consortium. In West Africa, in Abuja, Nigeria, in East Africa at Makerere University, and then in South Africa is the biorepository servicing Southern African region. This is just a picture of the biorepository, which is based in Johannesburg, and those are some very pretty -80 freezers, which are used for the HE Africa project. Now, as everybody is probably aware, we've had a pandemic in the world over the last 18 months. And this has brought biorepository science to the fore because we have started to question the value of the biorepository. And biorepositories can collect clinical material as well as allow for innovative biodiscovery for things like novel therapies, vaccines, and biomarkers for health and disease. This is not the first time that we've used biorepositories as part of the public health initiative. So a public health emergency of international concern was declared first for the Ebola outbreak in 2014 and 2015 in West Africa. And several biorepositories were developed for storage of material from individuals who had become infected. An example is the Ministry of Health and Sanitation, as well as Public Health England Biobank, which was in Sierra Leone, which collected material from approximately 1,100 infected individuals. There were some lessons from those biorepositories, however, with respect to the ethics. There were questions regarding the delays in ethics approval from the WHO, as well as country-specific bodies, and this was often related to poor quality submissions, queries related to vulnerable populations, for example, women and children. The need for templates for agreements became apparent, as well as the requirements of benefit sharing and the need to liaise with in-country populations. And this was reviewed quite recently in terms of what lessons were learned from the Ebola outbreak. So the Ebola outbreak was the first test case of the use of biorepositories in a public health emergency of international concern. And several biorepositories were established to collect material from individuals who had become infected. For example, the MOHS Public Health England Biobank in Sierra Leone. But there were some concerns related to these biobanks, including the delays in ethics approval from the WHO and in-country specific bodies, which is all often related to poor quality submissions, queries related to the collection of material from vulnerable populations like women and children, the apparent need for templates for agreements, for example, material transfer agreements and data transfer agreements, a clear policy regarding benefit sharing for in-country participants, and the need to liaise with in-country populations as well. The Ebola outbreak also provided a test case for the development of vaccines for this type of viral infection. And several ethical questions were raised with respect to the Ebola vaccine. The vaccine trial for Ebola, which was conducted in West Africa, prioritized healthcare workers who were assumed to be at high risk of contracting the illness, but did not include unregistered healthcare workers and community carers who might also be at risk of contracting the infection. There was a placebo arm for the vaccine trial, which means that there were some people who did not in fact receive the vaccine, and concerns were also arising about what was done with the various samples which were shipped across the world. So why were the HE Africa biorepositories ideally placed in this current crisis? So a number of the queries that were raised with respect to the Ebola crisis were actually addressed prior to 2014 by our policies. We had policies and procedures in place to guide the storage of material and templates for material and data transfer agreements. We had a significant infrastructural development, prior ethics approval, and extensive stakeholder engagement, which focused on those populations who were considered to be at risk as well. Finally, we have been in the process of formulating a coherent policy on benefit sharing as well. In South Africa, the first cases of SARS-CoV-2 were identified in early March, probably imported by tourists. And as of the 19th of August, South Africa has the highest number of cases in Africa, with approximately 2.6 million and 78,000 deaths. All three of the biorepositories began collections for their banks in March, and these samples were used for the evaluation and development of diagnostic strategies and various research applications as well. So in Johannesburg, we collected approximately 700 individuals who were convalescent for SARS-CoV-2 to establish a diagnostic panel for evaluation of both molecular and serological assays for SARS-CoV-2. The other benefit of having a well-curated biorepository was that we were able to also include negative controls within these panels that had been stored prior to 2019. And these included patients with viral pneumonias of additional origins, so not just SARS-CoV. This has been really a very fruitful interaction. We have published extensively on serological validation, as well as the impact of SARS-CoV-2 on patient health. In addition, we've collaborated with a number of researchers, both in South Africa and in the continent at large, and for example, using novel technology to develop SARS-CoV-2 antigens in plant-based systems as well. As of now, when I'm speaking, we have validated 5 formal and 30 point-of-care assays for SARS-CoV-2. Into the future, I think this collection is going to be extremely important for innovative and locally made diagnostics, external quality assurance panels, and for vaccine research. And we have been extensively involved with vaccine development in South Africa. As you can see here, we've been involved in both 2 big seroprevalence studies, the SAMRC and HSRC, as well as 2 vaccine trials in South Africa, the Novavax and the Pfizer vaccines, as well as an innovative award from iLead to look at novel therapeutic development. So we're thinking that the biorepository has really played an important role in public health response in South Africa and in Africa. I'd just like to thank my staff in the biorepository, especially Mukta and Garth, my staff in the Department of Immunology, especially Mayemu, Sarika Nakampi, my collaborators in molecular medicine and hematology, and the National Health Laboratory Service, and you for your attention. Thank you.
Tim Moshoma, who's going to talk about room temperature nucleic acid storage. Tim.
Good day. My name is Timothy Moshoma. I'm a master's student at the University of Witwatersrand, Johannesburg, and I'll be presenting about my project, which is titled Optimization of Ambient Temperature Storage of Nucleic Acids. H3 Africa is a funded initiative which aims to study the African genetic determinants of disease while increasing capacity for high-quality genetic research. Infrastructural resources have been built with biorepositories located in Nigeria, Uganda, and Johannesburg, South Africa. The main importance of repositories is to store aliquots of genetic material from research projects across various countries. Biorepositories have assisted participation in international arena, biorepository scientific research, and driving related policies, especially those relating to biorepositories ethics. CLS has a biorepository that stores a variety of samples at low and ultra-low temperatures. There are several methods for assessing DNA and RNA concentration and its purity. Some of the methods include spectrophotometry. In our lab, we're using the nanodrop. So the advantage of using the nanodrop is that it's rapid and you can measure low nucleic acid concentrations, and low sample volumes are required. The disadvantage is that it can overestimate the concentration of DNA and RNA. There's also gel electrophoresis, which gives an accurate evaluation of the integrity of our nucleic acids. The issue is that it's time consuming and it's a manual method. DNA and RNA integrity number, the advantage is that it's automated, standardized, and the results can be reproducible. The issue is that it can be technically complex. We also have PCR. So with PCR, the advantage is that you can use small concentrations, with the disadvantage being that it requires accurate calibration and validations, and it's also time consuming. Maintaining nucleic acid integrity is a challenge in areas with limited resources. Constraints include power supply issues, infrastructure for processing and maintaining the cold chain, storage capacity, transportation, especially across borders. So nucleic acids can degrade, especially when transfer times to laboratories are delayed or if the samples are subjected to extreme temperatures. A number of stabilizers exist to optimize nucleic acid storage. In addition, our center has pioneered DBS for a number of applications, including HLA typing. The aim of this study is to optimize ambient storage solutions for nucleic acids at the H3 Africa Clinical Laboratory Service biorepository, with the major objectives being to optimize DNA and RNA extraction for sequencing applications using a manual and an automated method, to evaluate different storage solutions for DNA and RNA at ambient storage, to develop a protocol accurately to assess DNA and RNA quality post-storage. 8 ADTA samples containing whole blood submitted for full blood counts at clinical laboratory services were collected. The ethical waiver was obtained from the Human Ethics Research Committee of University of Witwatersrand. So the ADTA samples were extracted for DNA using the Hamilton instrument, and they were extracted for RNA using the RNA Easy RNA extraction blood kit. After extraction, DNA integrity was tested using gel electrophoresis, and DNA and RNA concentration were tested using the nanodrop. The extracted DNA and RNA was stored in DNA and RNA storage solution and adsorption beads. And alucodes were then placed at different storage temperature conditions for 24 hours and 96 hours. After each time point, DNA and RNA concentration and DNA integrity were tested. Samples were also spotted on the DBS cards and stored at different storage temperature conditions for 24 hours and 96 hours. RNA Easy RNA extraction blood kit was used to extract RNA, and the concentration tested using the nanodrop. DNA concentration for alucode stored at 40 to 50 degrees Celsius was highly decreased after 24 hours and 96 hours. Alucode stored at lab room temperature had DNA concentration slightly similar to DNA concentrations of alucode stored at 2 to 8 and minus 80 degrees Celsius for both 24 hours and 96 hours. Gel electrophoresis showed that the initial DNA integrity of all samples was good, as all samples had thick, bright bands. You can see for all samples, the bands are thick and they're bright. After 24 hours, alucode stored at 40 to 50 degrees Celsius showed to have degraded DNA, as there was faint smeared bands. Similar results were obtained for alucode stored for 96 hours. You can see here, even at 96 hours, the bands are smeared and they are faint. Alucode stored at 40 to 50 after 24 hours and 96 hours had decreased RNA concentrations. This is a similar pattern as seen in the DNA concentrations. And similarly to DNA concentrations, RNA concentrations of alucode stored at room temperature had slightly similar concentrations towards alucodes stored at 2 to 8 and minus 80 degrees Celsius. RNA concentrations from DBS for 24 hours and 96 hours were lowest at temperatures of 40 to 50 degrees Celsius for 24 hours and 96 hours, and remained high for DBS stored at room temperature. You can see at room temperature, the concentrations were even slightly similar to the initial concentrations, and the ones stored at 2 to 8 and minus 80 degrees Celsius. Nanodrop and gel electrophoresis results support that DNA and RNA stored in different support medium remain stable at room temperature. The results also show that nucleic acids are stable for more than 96 hours at room temperature. Thus, we consider room temperature storage technologies as an alternative and cost-effective to cold chain logistics. Further testing is underway to check if the stored DNA and RNA can be used for downstream application. And this further testing include housekeeping gene quantitative PCR and HLA typing sequencing. I would like to acknowledge the National Institute of Health and H3 Africa for funding this project, the National Health Laboratory Services, University of Witwatersrand, Vet Health Consortium, Clinical Laboratory Services, H3 Africa staff, and I would also like to thank my supervisors, Professor Elizabeth Main, Dr. Maimu Gedeza, and Mr. Nakambe Mampeule for being supportive throughout this whole project. I thank you.
Well, thank you to Tim for that fascinating talk. We're now going to move to Uganda and hear about the Integrated Biorepository project there. And this will be presented by Moses Jaloba.
This is Moses Joroba making a presentation about the Integrated Biorepository of H3 Africa Uganda, and we are going to provide an update. These are our project milestones to continue to offer biorepository services to H3 Africa projects and other researchers, expand scope of biospecimen, consolidation of sustainability plan, respond to current and emerging diseases such as COVID-19. In our milestone to continue to offer biorepository services to H3 Africa project and other researchers, this table summarizes our engagement for phase 2, where we have received samples from Carfugen Uganda, plasma, PBMC, DNA, RNA. We have processed, stored, and shipped some. We are expecting samples from Carfugen Botswana, also from Trypanogen, TB gene in Ethiopia, and PTSD from Rwanda. This table summarizes what we expect. In addition, we have gone into networking, such as participation in H3 Africa Consortium meetings. We have continued to attend and participate in these meetings with our students. We have also offered training in biobanking services in Uganda and other areas. We do courses and course units in biobanking science, bioinformatics, and molecular biology. Also support for research. We offer research platform for local and international PIs, researchers, and students. In manuscript writing and publications, we have written manuscripts with other H3 Africa biobanks, such as measuring utilization and sustainability, site engagement with H3 Africa biorepositories, and actually multiple manuscripts from students and investigators utilizing biospecimens from our biorepositories have been written and published. We have expanded the scope of biospecimen, such as venturing into advancement into tissue and organ biobanking. We have set up a bank for sperm and ova, and also for stem cells harvested from cord blood, as shown here in this upper panel. But we have also done community engagement on ethical, legal, and social implications of organ and tissue biobanking and transplantation. The picture here shows experts engaging media and the population about organ and tissue biobanking. We have also expanded the scope to include environmental samples. We have developed protocols for collection, processing, and storage of samples from environmental exposures. We have collected and stored the environmental samples from a number of studies, such as silicone bracelets, urine, plasma, soil, leaves, and filters. We are in contact with a collaborator interested in studying pollutants in organ blood and its effects on mothers and children in Uganda. So cartoon here shows air sampling filter after 24 hours of household air pollution monitoring. We have built capacity in environmental samples. The team has just completed training in HPLC techniques in the process of and we are in the process of equipping the biobank with HPLC MC systems. And also we have done grant application in environmental exposure. We have applied to here around 6. Unfortunately, we missed funding, but we have received very positive comments and planning to reapply to here around 8. We have also applied for Government of Uganda funding to analyze heavy metal exposures. Part of sustainability plan implementation, we are now managed by Makerere University Biomedical Research Center, BRC, which is a semi-autonomous center of excellence under Makerere University, actually summarized here in this panel. And this center is a self-accounting, which comes with the ease of human resource management, procurement, and resource mobilization. Under the BRC, IBRA is able to engage different stakeholders in resource mobilization. And we think this is really very key for our sustainability. Also in sustainability, biorepository is a key resource to several projects outside H3 Africa Consortium. The table here shows the various studies we engage with their PIs, and we are running accounts for them, and different researchers pay a fee for service in terms of cost recovery, which is a key also for sustainability planning. And we continue to engage many other researchers and also support students to utilize stored biospecimens to answer new questions. Quality assurance is very key as part of our sustainability plan, and we do biospecimen quality monitoring using internal quality assurance programs and continue to participate in external quality assurance proficiency test by IBPL. And I'm very happy to also tell you now we have enrolled for ISO 20387 biobanking program, and we are on the road to accreditation. And we think this is really very key part of our sustainability plan. We have secured local funding as was part of the plan for the overall H3 Africa Consortium. We have successfully applied for funding for 2 grants from Makerere Research Innovation Fund, and also received funding from Government of Uganda to set up a COVID-19 repository to support research here. We have also received funding from our government to set up an innovation hub for diagnostic in the country. This is a factory which will be able to produce products. And here is our architecture plan for that factory. And we have increasingly positioned ourselves as a key resource in Uganda. And we have been noticed now that actually the government appreciates our role in biotechnology. In response to COVID-19, we successfully set up a COVID bank, fondly called as COVID Bank, with well-annotated COVID-19 positive and negative samples. That way, we have supported research groups and biotech companies with archived samples, and also participated in nationwide COVID-19 routine testing and participated in nationwide random assessment surveillance for COVID-19. Here, this graph shows that Makerere University, our lab, heavily participates in COVID testing. Actually, we are third in terms of volume in the country, which is a very significant contribution. We have also aided the development of COVID-19 technologies using our COVID bank, such as RDT kit development, laboratory validation, and field validation. The cartoon here shows how our samples from our COVID bank have supported biotech companies, vaccine, diagnostic development, product development, and many more samples. So we have been contributing heavily in that regard. In terms of sequence, using our COVID bank, we have performed the sequencing from the stored samples, and we will continue to sequence and contribute to Ministry of Health surveillance for COVID-19 new and emerging variants. This shows our catalog and our URL. You are invited to visit, show the specimens we have, and you can interact with us. Thank you, and thank you very much. I'm going too fast. Stop recording.
Thank you very much. Well, we're now going to do a geographical shift to West Africa, and for our final part of this session, and we're going to hear first from Alashley Abimiku.
Good afternoon, colleagues. My name is Alashley Abimiku. I'm the principal investigator for the Institute of Human Virology, H3 Africa biorepository that supports projects across West Africa. With me today is the biorepository manager, Mrs. Petrolina Ozumba. So I'll present on the first few slides, and Petrolina will provide more details. Just to remind everybody, about 9 years ago, the Institute of Human Virology biorepository was funded by the National Institute of Health to build its capacity to support multiple H3 Africa investigators across West African countries and the wider scientific community to conduct high quality genomics research, as well as translational research using well-processed, preserved and quality control biospecimens that were collected on the appropriate ethical approval. So just to give you a summary of where we are at the moment, for the first 5 years, we were funded to support about 5 to 6 different projects across West Africa. All of the biospecimens from this projects have all been deposited within our biorepository and allocated and ready for distribution for further scientific work. For the second phase projects, we've got about 7 projects. Only 3 of those projects have had some traction in terms of depositing biological specimens. So obviously we'll be working with the other 4 projects to make sure that samples are deposited, especially as we come towards the end of the H3 Africa funding. In terms of data submission, we've worked really closely with Nikki Mulder and her team in standardizing and harmonizing the language and data that accompanies the biological specimens. And obviously that is ongoing for the phase 2 project for additional biological specimens that will be submitted. So this is an ongoing activity. So with that, I'll stop here and hand over to Petrolina Ozumba to give you more details for all of the summary presentation that I've provided. Thank you for listening, but please stay on for Petrolina's presentation. Thank you.
Good afternoon. As Prof mentioned, I am the IHAB Biorepository Manager. My name is Petronila Ozumba. I have implemented 6 specific aims. Aims 1 and 2 with focus on quality. I have continued to maintain quality and also operate at international standards through these activities. We've been able to complete 2 audits by the Federal Ministry of Health and 1 from IHAGREN Quality Assurance Units. We also completed 1 ISBA self-assessment test and obtained a 98% score, which is an improvement from the previous 95 of last year. We've continued to enroll and participate in annual proficiency testing. We participate in 2 schemes, DNA extraction and QC, and obtained very satisfactory reports in 2020. I have also conducted retests of all samples that are deposited. Based on percentage concordance, we retest either 10 or 100%. We've tried to automate some processes to reduce human error and also increase efficiency. For aims 3 and 4, catalog and redistribution, we've continued to work with the H3 Africa BioNet to populate the online catalog. And we've also initiated processes to pilot sample distribution. We've automated sample management and have utilized freezer works to track samples and deposits. For aims 5 and 6, that focuses on sustainability. I have incorporated best practices and ethics into biorepository functions. And we've also continued to explore on the established long-term models for sustainability. This will be discussed in details in subsequent slides. Furthermore, our community engagement, we've been able to print instructional and educational materials and have also been able to upload a video on the YouTube. I would encourage participants at this call to please view the video. We've also continued to upgrade the standards of our staff, and through trainings, we were able to complete 8 trainings that spanned across project management, risk assessment, good laboratory practice, scientific writing. In terms of sample deposits, recall part of IHAB's deliverable is to secure sample deposits from H3 Africa supported projects. For phase 2, 3 of these projects have made some progress, and these are the Beaming project, the Eyes of Africa project, the Abricane projects. For 2 other projects, the NEETs in Mali and the Sickle Gen Africa in Ghana, we expect sample shipment from them. Initially, we had scheduled shipment in August, but this didn't happen. So I'm going to be following up with the 2 peers for the NEETs project Mali and Sickle Gen Africa, including the Kidney Disease Research Network Ghana, to ensure that the samples are secured and deposited at the biorepository. So despite the challenges with the COVID-19 pandemic, we've also moved forward in terms of our publications, and we also took advantage of the pandemic to support COVID-19 related studies. Under the manuscripts, I have published 1 publication. We have 1 that we are currently drafting, and we have 4 that we are drafting with our sister biorepositories. For COVID-19 support, IHAB is supporting the evaluation of rapid diagnostic test kits to develop testing algorithm for SARS-CoV-2 infection in Nigeria. We're supporting 2 of these evaluations. Then we're also supporting 2 clinical trial studies and 1 seroprevalence study under the Secure Nigeria grant. As I mentioned, just like the rest of the world, IHAB was also impacted by the COVID pandemic. The impact was mainly on three areas: the operations, processes, and logistics. IHAB set up a committee, COVID-19 committee, to track the disease among staff and also vaccine intake. We had to, because of lockdown, we had to initiate a remote work schedule and also staggered physical work schedule to maintain physical distancing as required. The staff were also cross-trained to back up activities. For the processes, as I've mentioned earlier, we had to support a lot of COVID-19 research. We received a lot of requests, and we had to expand our services specifically to support the vaccine storage and also processing of viable cells. For logistics, this is the area that was most affected. We had a lot of back orders, cancellations, and we had to identify local sources as alternative sources for our supplies. We had to even reschedule some sample shipment and deposits due to border closure. For the future, we intend to continue to explore diverse funding sources. We'll utilize our institutional support, our fee-for-service model, and also support collaborative grant applications through our International Research Center of Excellence. We'll continue to expand our services and currently are providing support to the government of Nigeria. That includes working with CDC, the Federal Ministry of Health, specifically to set up biorepository campus under a cost recovery arrangement. We intend to leverage on our newly established liquid nitrogen plant at our new sites to expand services to non-HA Africa and also target the academia. We'll leverage on experiences with COVID-19 and the response to also serve as resource for future epidemics and pandemics. So finally, I would like to thank my PI, Professor Lashley Abimiku, for her leadership, the IHAB team that worked tirelessly, irrespective and despite the pandemic, the HA Africa Consortium, and our sponsor, the NIH. Thank you. Please continue to remain safe. Bye-bye.
Well, thank you so much to all the presenters on biorepositories. We now have just a few minutes for questions, and I will go through them. The first one is for Tim, and it says, Tim, thank you for the nice presentation. Are there any related experiments that looked if the storage and repeated freeze and thaw of blood samples eventually affects the quality of RNA and DNA? Tim, are you there?
Yes, I'm here.
Right. Did you hear the question?
All right. Yes, I did hear the question.
Right.
All right. Thank you for the question. Well, to answer the question, I would say we haven't really looked at the freeze and thaw effect on the samples, like looking at doing an experiment of some sort. But through my observation in the lab, I've noticed that for RNA, since it degrades quickly, freeze and thaw times, freezing and thawing for close to 5 times, it actually kind of like degrades your RNA, because I've seen that the RNA concentrations would go down, and also on the gel you'd not see like perfect, I would say, bands. And also for DNA, it was roughly close to, I would say, 10 freeze and thaw cycles. but I haven't really done sort of like a related, experiment on that.
Okay, thank you, Tim.
So yeah, thank you.
The next question is for all of you, and it's a question about the difference that H3 Africa has made to your support, to your capacity to support biomedical research, and whether your institutions and or governments recognize the effort and the input and the advances in this area. Who would like to start off? Ashley.
Hi. No, without a doubt, it has made a significant difference in even understanding what biorepository is all about. A number of the documents that we have put together, because we have worked across Africa and gotten the best practices, has really helped what we do. The processes, the engagement of shipping, courier services, and all of that. I mean, 10 years ago, that was a nightmare. But right now, H3 Africa has really helped in testing all of the systems across West Africa, across South Africa, across East Africa. And now H3 Africa has these documents that you can actually engage shippers and researchers and government with. Certainly one clear example I can give from the infrastructure we set up in Nigeria is the fact that for the first time, H3 Africa, a national biorepository, the National Ethics Committee has the process of certifying biorepositories, in which case they go in, they look at your processes and how you engage communities and all of that. So without a doubt at all, in terms of collaborating, in terms of having ISBER best practices, the resources, the infrastructure, the knowledge, the community engagement, and all of that, it wouldn't have been possible. And then being able to work with sister biorepositories across South Africa and East Africa has really just expounded a lot of the research that we are able to do. And as investigators ourselves, it's really helped the ability to share.
That's very good to hear. Thank you. I'm sure other people would have comments to make about this, but I think we need to go on to the last question, which is a question that's often asked about biorepositories, and that's about sustainability. So there's a question about what plans there are for sustainability or what prospects. Moses, would you like to take this one?
Yeah, thank you very much. I hope you could hear me. Yeah, that's really a very big question, which I always it's very tough to answer. But I always argue that if you succeed, it is very easy to sustain. Now that we have really, as my colleague Ashley Hebley illustrated what successes we have achieved and what we have presented, and now that people are aware of the good things the biorepository can offer, there is a good chance that we will sustain. We have training, the biorepositories are embedded in institutions, we have been able to collaborate and build networks with the biorepositories and other researchers, and governments have recognized us. So I think the biorepositories are here to stay. Thank you very much.
And thank you very much. And thanks to all the presenters and the attendees and those who've posted questions. And I'm particularly pleased that we've finished in good time at this plenary session. And it just remains for me to announce that immediately there's going to be a meeting for health librarians, as well as a meeting of the IEC. So we'll wrap this up now, and thank you once again.